Hi, my name is Jovana Mogmirovic. I'm doing the Bachelor of Computer Science, and welcome to my thesis presentation about my research titled A Fifth-Order Adaptive Network Model of the Role of Epigenetic Modification Caused by Early Life Stressors in Borderline Personality Disorder. And I'd also like to thank my supervisor, Jan Treur, for supervising me these past couple of months. So the addressed research question is, how effectively can the role of epigenetics in BPD and some therapies for it be modeled using a fifth-order adaptive network model? And some context for this research question is that epigenetics play a significant role in mental disorders, and the interactions between the environment, epigenetic modifications, and the subsequent development of the disorder are complex, dynamic, and adaptive interactions, so it would make sense to use an adaptive model. And it's important to investigate and analyze these interactions to potentially improve our understanding and treatment of BPD. So according to the DSM-5, the diagnostic criteria for the disorder are instability in several domains, namely affect regulation, impulse control, identity, and interpersonal relationships. Besides that, intense emotions and stress seem to be central to other symptoms and coping mechanisms of disorder, such as self-harming behavior or substance abuse. And stress-related dissociative experiences are also frequently reported in patients with BPD. And there seems to be a strong association between childhood maltreatment and a disorder. So epigenetic processes are processes that affect the gene expression without changing the DNA sequence itself. These processes can be influenced by environmental factors, and childhood is one of the susceptibility periods in which epigenetic programming is sensitive to environmental stressors. And DNA methylation, which is an epigenetic process, is believed to decrease gene expression in most genes. So specifically for BPD, studies have found an association between increased methylation of the NR3C1 gene, so hypermethylation of this gene, childhood maltreatment, and the development and the severity of the disorder. So this NR3C1 gene, also called the GR gene, encodes the glucocorticoid receptor protein. And this protein binds to the stress hormone cortisol, which is produced by the HPA axis. And this binding acts as negative feedback to the HPA axis. So the HPA axis is the hypothalamic pituitary adrenal axis, which regulates the neuroendocrine stress response of the body. In the figure, you can see the schematic representation of this axis. And the important thing is that cortisol is one of the end products of this axis. And then GRs, this protein, is expressed throughout the whole brain. But there are certain areas with a higher concentration, making them the main inhibitors of the HPA axis. And these are the PVN, which is located in the HPA axis itself, the hippocampus, and the prefrontal cortex. And the amygdala is thought to be the main activator of the HPA axis. So what are the specific effects of this epigenetic change? Well, hypermethylation of the GR gene leads to decreased negative feedback to the HPA axis because GRs are less available to bind to cortisol, and so this leads to increased cortisol levels. And prolonged exposure to cortisol leads to a constant state of hyperarousal, triggering the fight or flight response. And this also leads to abnormal nervous system activity and overstimulation of the amygdala, hippocampus, and prefrontal cortex. And this in turn also leads to inappropriate behavioral stress responses. And this figure also shows these behavioral stress responses, such as misinterpretation of perceived signals from the environment as constant threats, unstable and intense emotions, and cognitive dysfunction, such as loss of decision-making capabilities and rationality. So the modeling approach that is used for design models is the adaptive temporocausal network modeling approach introduced by Treur. And these models consist of network nodes or states that are dynamic with their own values and network characteristics. And these characteristics are consist of connectivity. So they include directed connections between states that indicate a causal relation between them with weight omega. You have the aggregation characteristic, where the combination function C specifies how combined causal impact of incoming connections to the state is calculated. And you have the timing characteristic, where the speed vector eta determines how quickly a state Y adjusts its value after receiving the aggregated causal impact. And here you can see the equation that is used for the dynamics of a model, which includes the network characteristics that I just mentioned, like eta, C, and omega. So what I just explained gives you a dynamic model, but to create an adaptive model, you can make the network characteristics omega, C, and eta adaptive by adding self-model states representing these characteristics. And these states are placed at higher network levels above the base level. So for example, self-model state W are used to represent adaptive connection weights omega. And this principle can be applied iteratively throughout higher network levels by adding second and higher order self-model states. And this is also used in the design model. And later for the simulation experiments that you'll see, I use the combination functions advanced logistic sum and step ones, each with their own parameters as well. So now we get to the actual design models. So this is the main model that represents the development of BPD. And on the base level, you can see the stress regulation, where a person senses a stress stimulus and then the body prepares itself for a stress response. And this stress response is suppressed by the GR state, which has a negative connection to the HPA state. And then on the first self-model level, we have the brain structure that concerns the connections between the neurons. And on the second self-model level, we have the production of the GR protein. On the third level, we have the production of messenger RNA that is used for the synthesis of the GR protein. And on the fourth level, we have the expression of the GR gene. And on the fifth level, we have the level that represents the epigenetic change. And then all these W states on the higher levels, they affect the availability of the GRs on the base level. Then the second model is the model with the epigenetic therapy, where the epigenetic therapy is placed on the fifth level. So con-TSA is the state representing the epigenetic therapy. And this is a hypothetical epigenetic therapy using trichostatin A, because this has been found to demethylate the GR gene in adult rats. So theoretically, it could also be used in patients with BPD to demethylate the GR gene in a hypothetical epigenetic therapy. Then the third model incorporates dialectical behavior therapy, which was introduced by Linehan and involves skill training and enhancing problem-solving skills in people with BPD. And it is thought to lower the activity of the amygdala, but it is a non-epigenetic therapy, so it doesn't address the cause, the epigenetic issue. And that's also why it is placed on the base level with a connection to the amygdala and not on the epigenetic level, like the trichostatin A from before. So the first simulation is from the first model, showing the development of BPD and the effect of the epigenetic change. And in the simulation on the left, it can be seen that before the epigenetic change, so until time 150, when only the regular stress stimulus is present, the base level states such as the HPA state, the cortisol level, the amygdala, prefrontal cortex, and hippocampus are all relatively low. And this is also because the suppression by GR still works as intended. And it's also shown by the W states that are still active in the figure on the right. But between time 150 and 280, the early life stressor is present and the hypermethylation of the GR gene takes place. This can also be seen on the right, where the W states all go down and eventually reach zero, representing the dysfunction of the GR protein production. And so after this epigenetic change, after time 180, states representing the amygdala, prefrontal cortex, hippocampus, but also the HPA axis, cortisol level, and nervous system are very much active compared to before the epigenetic change. And this represents the hyperarousal of the body and subsequent behavioral stress responses seen in BPD. and so this signifies the persistence of the epigenetic change in the symptoms, even when the early life stressor is not present anymore. So the second simulation is from the second model, so the epigenetic therapy model, and between time 400 and 530, the trigostatin A is administered, and thus the GRs become active again. It's also shown by the W states going up again on the right. And we see that after this demethylation of the GR gene, the states that were previously highly active, such as the HPA axis, the cortisol, and the brain regions that we discussed, now go down again as a result of the epigenetic therapy, and actually reach levels that are similar to that of a healthy individual, comparable to before the whole epigenetic change in early life stressor took place between 150 and 280. So in this last simulation, we have the third model, so the model with dialectical behavior therapy, and DBT takes place between time 400 and time 530. And we see that because DBT has an effect on the activity of the amygdala, state representing the amygdala goes down, and this also has an effect on the other base level states that are involved in stress regulation. But because this therapy doesn't address the cause, the epigenetic change, when DBT is over, these states all go up again, and the individual still suffers from the BPD symptoms. And this can also be seen in the figure on the right, where the W states do not go up during DBT and also after. So the production of the GR protein is still not normal, and the individual still suffers from these symptoms. Then lastly, we have the discussion. Well, based on the simulations that we just saw, it could be said that the models successfully describe the effects of hypermethylation of the GR gene and two forms of therapy on borderline symptomatology based on the discussed literature. But there are also some limitations. For example, the model could be extended with other genes that are also implicated in BPD, namely DRD2, 5-HTR3A, BDNF, MAOA, and soluble COMT. And also, one thing is that the research on the epigenetics of BPD is still in its early stages, and it's actually been fairly inconclusive so far. So it's a bit difficult to draw general conclusions with certainty, and this is just something to take note of. And also something that complicates the research into BPD is the high prevalence of comorbidity. For example, PTSD, major depressive disorder, and bipolar disorder are frequently comorbid with BPD. And so sometimes it's difficult to find out if certain findings are exclusively related to BPD or also to other comorbid disorders.